It's Wednesday, the 22nd of March, 2022.
Thanks for joining me here today. Now, today's lecture.
So in today's lecture, we'll look at 7 key core concepts. Now, the first concept, we'll have a look at the different types of microscopes and the biochemical tools used for cell research by biologists. In key concept number 2, we'll look at eukaryotic cells and how their internal membranes compartmentalize their functions. In key concept number 3, we'll have a look at the eukaryotic cells' genetic instructions, where they're housed and how these instructions are carried out by the ribosomes inside the cell. In concept 4, we'll explore the endomembrane system and how it regulates protein traffic and performs metabolic functions in the cell. In concept 5, we'll look at mitochondria and chloroplasts and how they change energy from one form to another. In concept 6, we'll look at the cytoskeleton and how it organizes the structures and activities in the cell. And finally, we'll finish today's lecture by looking at the extracellular components and the connections between cells and how they help coordinate cellular activities. So we're going to have a quick look at the fundamental units of life, not the fundamentals of life, but the units of life. And the cell is as fundamental to the living systems of biology as the atom is to chemistry. All organisms are made of cells. In the hierarchy of biological organization, the cell is the simplest collection of matter that can live. Indeed, there are diverse forms of life existing as single-celled organisms. More complex organisms, including plants and animals, are multicellular. Their bodies are cooperatives of many kinds of specialized cells that could not survive for long on their own. However, even when they are arranged into higher levels of organization, such as tissues and organs, cells are an organism's basic units of structure and function. One of the first instruments that you'll use as biologists to study cells is a light microscope. Now, in a light microscope, visible light is passed through the specimen and then through glass lenses. The lenses refract or bend the light in such a way that the image of the specimen is magnified as it is projected into the eye onto photographic film or onto your camera lens on your phone. Even though microscopes can look very different, they all use the same principles to magnify an object and have the same basic parts. So you can see from the slide that this light microscope has many different parts, such as the arm, the stage clips, coarse adjustment, the fine adjustment, and the ocular lens. Now, the primary magnification lenses are the objective lenses, and you can see here we have 3 of those. And these objective lenses allow the user to go from low magnification to high magnification once they have found an area of interest in their sample. Now, at very high magnifications, an objective lens may need to be paired with immersion oil, which helps minimize the bending of light to increase image resolution. Now, there are essentially 4 different types of light microscopy. There are the simple light microscopes that use a single lens to magnify an object but don't really reach a high magnification. And then there is the compound microscopes that use 2 sets of lenses, an objective lens and an eyepiece to produce images. And then there is the monocular microscopes that have 1 eyepiece. And then finally we have the binocular microscopes that have 2 eyepieces. Here is a light microscope image of E. coli bacteria using 100 times objective lens and oil immersion fluid. The size of these bacteria can range from about 1 to 2 microns. Now, the 3 important limiting factors of our microscopy are magnification, resolution, and contrast. Now, magnification is simply the ratio of an object's image size to its real size. Resolution is a measure of the clarity of the image, how clear it is. Essentially, it is the minimum distance two points can be separated and still be distinguished as 2 points. Now, the third important parameter in microscopy is contrast. Now, contrast accentuates differences in parts of the sample. In fact, most improvements in light microscopy in the last 100 years have involved new methods for enhancing contrast, such as staining or labeling cell components so that they stand out visually in the image. However, one of the disadvantages of most staining procedures is that the cells are required to be fixed. The cells must be set in a preserved medium such as a wax or a cross-link solution such as formaldehyde. Before we leave light microscopy, let's have a quick look at confocal microscopy. Now, confocal microscopy is a type of fluorescent light microscopy. And this type of microscopy uses a pinhole aperture within the microscope to eliminate out of focus light from a thick sample. Now, this creates a single plane of fluorescence in the image. Now, by capturing sharp images and many different planes, a three-dimensional reconstruction of the image is created. Now, this allows structures smaller than 10 to 20 nanometers to be distinguished. Now, you've probably used a microscope in school, maybe to observe the wings of an insect or get a closer look at a leaf you found in the playground. If so, then you'll know microscopes are used in the classroom to illuminate the surface of your subject of study. Now, light microscopes use transparent glass lenses to magnify the image of whatever you are observing. However, the size of the smallest features that we can distinguish under the light microscope is on the order of the wavelength of the light used. Visible light, which is the one our eyes are sensitive to, ranges between 390 and 700 nanometers. 1 nanometer is 1/1000th of a meter. This means that we cannot observe things that are smaller than a few hundred nanometers using our eyes or the visible light in the light microscope. With the advancement of science and technology, we realize there is a whole world of things that we can observe and study at smaller scales. For example, the size of atoms and molecules is just fractions of a nanometer. An electron microscope allows us to see at these small scales. Now, electron microscopes work by using an electron beam instead of visible light and an electron detector instead of our eyes. An electron beam allows us to see at very small scales because electrons can also behave as light. They have the properties of a wave with a wavelength that is much smaller than visible light, a few trillionths of a meter. With this wavelength, we can distinguish features down to a fraction of a nanometer. Let's explore the different types of electron microscopes, how they work, and some of their applications. Now, there are 3 types of electron microscope: the scanning electron microscope, the SEM, the transmission electron microscope, the TEM, and the scanning transmission electron microscope, the STEM. In a scanning electron microscope, or SEM, a beam of electrons scans the surface of a sample. You can see this in the figure. The electrons interact with the material in a way that triggers the emission of secondary electrons. These secondary electrons are captured by a detector, which forms an image of the surface of the sample. The direction of the emission of the secondary electrons depends on the orientation of the features of the surface. There, the image formed will reflect the characteristic feature of the region of the surface that was exposed to the electron beam. In a transmission electron microscope, or TEM, a beam of electrons hits a very thin sample, usually no more than 100 nanometers thick. The electrons are transmitted through the sample, as you can see in the figure, and after the sample, the electrons hit a fluorescent screen that forms an image with the electrons that were transmitted. You can better understand this process by imagining how a movie projector works. In a projector, you have a film that has the negative image that will be projected. The projector shines white light on the negative, and the light transmitted forms the image contained in the negative. On this slide, we can see a TEM profile of a thin section of a specimen. Here we see a section through a tracheal cell, revealing its ultrastructure. In preparing the TEM, some cilia were cut along their lengths, creating longitudinal sections, while other cilia were cut straight across, creating cross sections. Now, a scanning transmission electron microscope, or STEM, combines the capabilities of both an SEM and a TEM. The electron beam is transmitted across the sample to create an image, TEM, while it also scans a small region on the sample, SEM. The ability to scan the electrons allows the user to analyze the sample with various techniques, such as electron energy loss spectroscopy, or EELS, and energy dispersive X-ray, EDX spectroscopy, which are useful tools to understand the nature of the materials in the sample. So what about sample preparation? What must I do to a sample before using a microscope? Well, you must make it conductive. How do I do that? Well, you must coat it with a conducting material. It could be gold, it could be silver, it could be copper, it could be carbon. Besides the wavelength, what might limit the resolution of my image using the electron microscope? Well, that's easy. It's the quality of the coating, particularly the grain size of the coating material. Help! My electron microscope is broken. How can I obtain an image of my cells now? Well, that's easy. You can use an AFM. Well, what does AFM stand for? Atomic Force Microscopy. Then what will determine the resolution of my image? The size of the tip. There are many different sizes of tips that you can use with AFM, such as 1 or 2 nanometer, or 10 or 20 nanometer. Another useful technique for studying cell structure and function is cell fractionation. This takes cells apart and separates major organelles and other subcellular structures from one another. The piece of equipment that does this job is the centrifuge, which spins test tubes holding mixtures of disrupted cells at a series of increasing speeds. At each speed, the resulting force causes a subset of the cell components to settle to the bottom of the tube, forming a pellet. At lower speeds, the pellet consists of larger components, and at higher speeds result in a pellet with smaller components. Cell fractionation enables researchers to prepare specific cell components in bulk and identify their functions, a task not usually possible with intact cells. For example, on one of the cell fractions, biochemical tests show the presence of enzymes involved in cellular respiration, while electron microscopy revealed a large number of the organelles called mitochondria. Together, this data help biologists determine that mitochondria are the sites of cellular respiration. Biochemistry and cytology thus complement each other in correlating cell function with structure.